But today, I mean, we're going to talk about the olfactory bulb. So let me share the PowerPoint. Because I have so many windows open, sometimes it doesn't show up. So can you see it now? Let me go into mode. Okay, so we have 5 students. I know two students.
Dr. Hayar, sorry for interruption.
Okay.
Can you how can the students get access to the PowerPoint? Can you email it to me so that I can put it on Blackboard for them to have access?
Okay. Actually, send them a link to the old file. You remember I used to have it on my website.
Yeah.
Yes.
Yeah.
So it's the same link if you still have it, or you can Google NBDS 5106. If you Google that, you'll get the page.
Okay, no problem. Thank you so much. Thank you so much. It's
Okay.
so you can start now.
So the taste olfaction is now an interesting topic because, as you know, people who get COVID-19, they suffer from taste and smell problem. So although I don't have specific slide on this, I'm going to show you how this might occur. So, you know, the virus can access our body through different ports of entry. So the nose and the mouth is the main part, because that's why you have to cover them with a mask, because so when the virus is expelled by a infected patient, so these particles can spread into the air as aerosol, and so you'll get them into your nose and into the tongue, and this is where they start replicating and damaging the cell. So you get smell and also taste deficit. And also the problem is that the virus can go through the nerve and then infect the central nervous system by going from one neuron to another neuron. So from the olfactory, for example, receptor neuron, or the taste cell, the taste bud, so they can go through the nerve, cordia tympani, and then they go to the brainstem. And so this, they can infect, for example, the brainstem, if you know, there is a central control of the cardiovascular system. So this is where respiration and cardiovascular rate is regulated autonomously, which means even if we're sleeping, it's regulated spontaneously. And so if this center, for example, get infected, then the patient will stop breathing on their own. So, so that's why you have to ventilate and give air, so by mechanical ventilation. So that's why it's important to mask up and cover the nose and tongue, because this is a main port of entry of virus. But also, probably have seen that the virus can also infect the digestive system, and it can cause heart problem. So, and so the lung will be, when they get infected, they go like into extensive inflammation and produce a cytokine storm. And this is when the symptoms become severe. So uh, the so we perceive the flavor of food using by smelling and tasting. So this is very important for the quality of life. So COVID-19 patients, so they lose this two senses, but only transiently. So you're going to see that the taste bud actually can recover very quickly. For example, if you drink like very hot coffee and you burn your taste bud, will they recover? Yes, so it's the cell divide, and so they replace the damaged cell. Also, the nasal epithelium, if the virus cause inflammation and kill large part of the cell, you'll be able to regenerate the cell after 3 to 4 weeks. But there are some people, probably you heard, that they will experience some strange smell. So they can smell some burnt food or something that abnormal, that they're only the people who smell it. So it's like they have illusion about different smell. So after being infected with COVID-19. And this is because you're going to see that when the taste bud or the olfactory receptor neuron are damaged, so they can create some kind of chaotic distribution between the taste bud and the central nervous system. So the brain will not be able to recognize the same type of odor or taste. So it has to be like reprogrammed and rehabilitated. So the taste bud are located on top of the tongue, and they can be in front, in the back, but also it's not just the tongue that can taste the food. So you can have also taste bud on the different area of the mouth, top of the cavity of the mouth, bottom, below the tongue, and even inside the esophagus. You can have some taste bud. So even if you swallow something very quickly that doesn't go through the mouth, you'll be able to both smell it and taste it. So there is some kind of sporadic, taste bud located at lower density in the first part of the GI tract. Now, one thing you can see about the taste buds, so they are located into like membranes that are like convoluted, and this is why, because you need to increase the surface area. So the more area you have, the more likely you're going to taste better. You can amplify the taste response by increasing the area. So they come in different shapes. You have circumvallate, foliate, fungiform. And so you can see that each taste bud is like some kind of onions with a pore on top. And so the food, or the molecules has to dissolve in the solution and then access cilia. So this is where you have the taste bud cell. And so this is where they bind to receptor and they cause depolarization of the membrane, and then electrophysiological activity that will be transmitted via the nerve all the way to the brain. So there are different group of taste, at least 5: bitter, salty, sweet, umami, and sour. We're going to talk about this more. So bitter is like coffee, olive, the citrus peel. So ammonium are more bitter than salty. So salty is like sodium chloride, potassium chloride, lithium. So sour mean like acidic, and when you drink a soda drink or citric acid, so it causes this sour. But you see, the Coca-Cola, for example, is sweetened. So you have the acidic flavor, but also you have the high fructose syrup, which is very sweet. And so this override the sour taste. So, for example, those that drink alcohol has both like a sour or bitter taste and sweet taste. So, and if someone become alcoholic, maybe their brain will forget about the sour taste of the alcohol, but they will only have the sweet taste. So that's why they keep drinking, because they perceive alcohol as sweet. So when they become advanced addict to alcohol, so they will ignore that bad taste. So sweet, so the milk contain lactose. So when it's disaccharide, it's split into smaller molecule, galactose, fructose. Now umami has a savory or meaty taste, so it's like cheese, and so it can be represented by also the monosodium glutamate. Now many of the like snack, they add monosodium glutamate to it because it causes salivation. So it actually also activates the gland to generate saliva. So it tastes good if they add monosodium glutamate. So you see the taste bud, so it has different type of cells. Now there is the basal cell, so these are at the bottom, and there is light cell and dark cell. Now why we call them like this? Because so the light cell can be identified with immunocytochemistry against an antibody for gusducin. So this is a receptor proteins on the membrane that transduce the taste signal. So this is like here you have a horizontal cut across taste bud, and here you have a transverse cut. So you can see now the cell look round. So the light cell here are labeled in red, whereas the dark cell, so they are labeled with another antibody. And so you can see that they surround the red cell and they have cytoplasmic projection. So this is like the main difference. Let's make the pointer visible. So as I said, the taste bud are located at different places of the mouth, under the tongue, the soft palate, the nasopharynx, epiglottis. And so they have different shape. So now, so when you're going to have the exam, don't remember details. So I'm not testing you for memory. I'm testing you for understanding how it works. So the idea from this slide is to show that the taste buds are located all around the mouth, and so even in the nasopharynx. Now the receptor for the taste bud can fall into 3 categories, and so there are usually 2 subunits that have to assemble together to make a functional receptor. So you have T1R1 and T1R3, so they assemble together and they'll be able to detect amino acid. T1R2 and T1R3. So here you see the T1R3 is shared, and so this will detect mostly sugar. And then you have another category, T2R, which has a lot of different like gene sequence and like 30 different receptor. And these are usually for bitter taste. So now can all animal taste the same as us? Can they taste sugar, salt? So you can see, for example, cat cannot taste sugar. So even if you give them sweet, it's like for them like not tasty food. So basically, they more like meat. So they are carnivorous. So that's why for them sugar they don't taste it. So mice have an amino acid taste. Now mice are good models. So if you can do knockout of certain gene, maybe you can identify whether this gene, for example, is responsible for a particular taste. And so if we knock out the T1R1, for example, we see that the mice will not be able to recognize or taste umami. If we knock out the T1R2, they will not be able to taste sweet. If we knock out T1R3, now both umami and sweet will not be recognizable. So you can see there are specific receptors that are programmed to recognize a particular taste. Now, if we do a knockout of some of the gene, you can see that more than 3 categories of taste will be lost. Now, how the effect is translated from the sugar binding to the receptor to an effect inside the cell. So this receptor are G protein coupled, and so they are connected to a GQ. So we're going to learn more about G protein coupled receptor in the other course. And so GQ connect to an enzyme called phospholipase C. So this phospholipase C will take phosphoinositol phosphate 2 and convert it into diacylglycerol, DAG, and IP3. Now, diacylglycerol, it will activate protein kinase C. The IP3 that is generated, it binds to some receptor on the mitochondria or the plasmic reticulum, and it will lead to release of calcium from the cellular store. So calcium inside the cell will increase. So normally the mitochondria and the plasmic reticulum, they buffer calcium and they have as much calcium as extracellularly, like 2 millimolar. But inside the cell, the concentration of calcium is much lower, less than 1 micromolar. And so one signal to the cell will be to increase calcium intracellularly. So this is released from the intracellular store. Now, there are different brain regions involved in taste. First, so the taste bug, when they get stimulated, there is electrical activity generated in the taste cell, and the action potential propagate via the nerve all the way to the nucleus tractus solitarius. So sometimes we call it NST, nucleus solitarius of the solitarius tract, sometimes NTS, nucleus of the tractus solitarius. So they are the same thing. So we have 3 nerves, the 7, 9, and 10, that are important to transmit the taste signal, so all the way to the brainstem. And then from the NTS, So this is a very long nucleus along the brainstem, and it can connect with uh, other brain structure. So either the brainstem, vertical formation, or it can go upward. So the parabrachial nucleus. So the parabrachial nucleus is the nucleus that will decode test information, and so can tell whether this is umami, this is sour, this is sweet. And then, so it sends signal, of course, to other region of the brain, including the gustatory neocortex. Amygdala, for example, if something tastes bad, I mean, if you have an expired cheese, expired meat, you can directly tell it tastes bad, so it's too old, and you throw it away. So the amygdala is like the region of the brain where you have your emotion is expressed. So if something tastes bad, so it's yucky, you see facial expression, and it can also cause you to reflex to throw out, vomit. Now, a neuron within the NTS can be stimulated by several type of category of taste stimuli. So a neuron is not particularly dedicated to recognize one type of taste. So each neuron can respond differently to different type of taste. So here we have one neuron, the solitary tract, and we applied different solution to the tongue. So this neuron sometimes is activated by sodium chloride, by a little bit sodium nitrate. So it's more like salty. It recognizes calcium chloride, magnesium chloride, and sometimes it responds with a longer response, several seconds. Sometimes it's a short response, or it like accommodate very quickly, so it go back to control firing. Now, distilled water usually has no taste, but I will caution you here because sometimes if you give the mouse or rat, for example, cold water versus hot water, it tastes differently for them. So they can recognize also the temperature of the tasting. And also distilled water, the problem is that it has no ions to buffer. So sometimes it tastes a little bit acidic. So you can see here, I mean, this slide looks complicated, but the idea here is that you can group neuron according to their taste response. So here, for example, we have 29 neurons, and we can see that some neurons are more responsive to sweet. So here you see all the tastings that have been applied. And so other neurons, they are more responsive to salt, salty substance. You can see here salty substance, anything that has sodium or magnesium, potassium in it. Some other neurons are responsive more to acid and quinine. So basically, you can group or cluster neuron depending on their profile. But they are not like specific for a certain substance. They can be activated by different substance with different amount of response, different affinity. So here you see also in the CNS, the parabrachial nucleus, so you have some neurons that can react to several substances, some only to sucrose, some only to sodium. sum to all of them. And then so you have further processing of taste information via interneurons. And so this will lead to activation of motor neurons. So either it tastes good, then there is like a reflex. The mouse will ingest the food, it will salivate and swallow. Or if it is bad, so it can cause coughing, gapes, and so you try to exclude the food. So also it will show on our face if we like the food or we don't like it. Now this is an experiment that I have done myself. It's published in one article. And so the rodent, they have specific way of tasting the food or drinking water. You know, they lick, and they lick at very high frequency, like almost 9-10 hertz. So 10 times per second, they lick. So only when you have like fast camera and you look at each frame by frame, you can see that the tongue, so it go out of the mouth, grasp some of the water from the spout, and then swallow it inside. So this occurs in 133 milliseconds. So this means 8 times per second. So you can see the rat here, it start licking fast at faster rate, and then it slow down. So maybe, I mean, it get tired, so it has to relax, but there is this repetitive licking behavior, and usually the rat or mouse, they can recognize the taste from the first few lick. So the first few lick will tell them whether it's good or bad, so they will keep on. So here, for example, it's sucrose. With sucrose, they continue to lick 300 lick in a burst. Whereas if you give them alcohol, which I mean tastes sour, so they will lick like 3 times, 4 times, and then they will stop. So their brain tells them, oh, that's not good, let's stop. So this is talking here about naive rat, not rats that have been addicted to alcohol. So those that addict to alcohol, they probably will keep licking the alcohol for longer time. So this slide shows you that the rodent, they lick repetitively at fast rate, and for the first few licks, they can recognize the taste. If it's good, they keep ingesting. If it's not good, they stop. I mean, of course, human cannot lick at this fast rate. I mean, it's weird, it's much slower. Now we move to the olfactory system. So in a human, the area in the brain responsible for olfaction are very tiny. So it's represented by the first cranial nerve. And so you can see there are receptors in the nasal cavity, so nasal epithelium. So this is where the coronavirus attach to this layer, and so it start replicating and damaging the layer, and so you lose the sense of smell. And you can see also that so the you have here olfactory receptor neuron, but they are connected to the brain via this olfactory nerve axon. And this, to go to the brain, they have to cross the bone. And to cross the bone, there are pores in the bone. So we call this the cribriform plate. So if you do gross anatomy and dissect, you'll see that this is, or you look at a skeleton, the skull, you will see that it's porous, there are holes. So this is where the nerve penetrate and they connect to the olfactory nerve. So this means that here, these pores are actually the pore of entry of pathogen into the brain. So any virus, any bad chemicals would likely be absorbed by the neurons go through the cytoskeleton and release from the axon terminal, then maybe infecting other neurons. So although the taste, sorry, the olfaction is not important for survival, so it was the subject of a Nobel Prize. So there's two people that got Nobel Prize for discovering the G proteins that are involved in the smell. And so these are the olfactory receptor that transduce the signal from odorant into electrical activity that transmit to the brain. So I said, I mean, for mice, olfaction is very important. For human, it's you can survive, but it will reduce the quality of life and might induce depression. So the animal are more sophisticated in terms of trying to identify and recognize odor. So you see, like dogs, or they can be trained to identify millions type of odors, and so they can sniff illicit drug, TNT at the airport, for example. They can be used trained to recognize even very small concentration of an odor. So you can see the human has can recognize odor and can also with the nostril, right and left. It's like, you know, your ear. You can tell where the sound is coming from. You can also tell from where the odor is coming from. Is it right or left? And then direct yourself toward finding that odor. So this is a human being trying to mimic like a pheasant and trying to look for a chocolate scent. So how do we know that olfaction is important in human, in rat or mice? Just dissect their brain, and you see that the olfactory bulb is almost the same size as the cerebellum. So it has a lot of neurons that are involved in the detection, recognition, and processing of olfactory information. And whereas in human, as I showed you in the previous slide, you can see that olfactory bulb is a very slender nerve on the base of the brain. So for animals, especially rodents, so they are nocturnal animals. They're guided mostly by odor at night to look for food. And another important thing is that it's important for reproduction and maternal function. So you can see, I don't know if you handled some rat or mice pups, the newborn. They actually are, their eyes are not open. They don't have also their ear open. So the only way a newborn mouse or rat can find his mother or is by odor. So they smell the milk and they go and try to get their mother and feed. So I said, so this is a port of entry of toxic chemicals, and there are some theory, I mean, still not well proven, that Alzheimer and Parkinson. So in both of these two diseases, the first sign or symptoms is a smell deficit. So which means, so this is before you develop Alzheimer. So you can screen a patient for possible Alzheimer by asking them to smell different type of odor. And if they have smell deficit, this means they're more likely to have some kind of brain damage already. And so this probably will take several years, and then they will start losing their memory. They have Alzheimer. So both Parkinson and Alzheimer can be predicted by smell deficit. So if you smell good, this means your brain is intact. But I mean, COVID can kill the sense of smell, but only in a transient way. It regenerates. So this is the anatomy of nasal epithelium. So it can be divided into different zones, different turbinate. So and so just to show you how it looks like, and there is one thing important here is called the vomeronasal organ. So this an organ that can detect pheromone. So you know, in insect and animals, so pheromone are also very important. So what's the difference between a pheromone and a real odor? So actually you don't smell a pheromone, you just react to it. So it doesn't have a smell, but it can induce reaction in so other individuals. So the olfactory bulb, so it's at the front of the brain. Sometimes if you look at the atlas, people usually ignore it because, I mean, they just look at the main brain to see what's wrong, the cortex, hippocampus. But the olfactory bulb is very important in animal. And so you have the main olfactory bulb and you have the accessory olfactory bulb. So the accessory olfactory bulb is the one that process the pheromone recognition. Then you have the accessory olfactory nucleus, the lateral olfactory tract. So this is our nerve that, so the output of the olfactory bulb go via nerve and it converges onto the piriform cortex, part A and part anterior, posterior. So these are the main region in the brain that process olfactory input. Now, an important principle in olfaction that can help you understand why an animal can recognize like millions of odor, we call this a combinatorial code. So there is about like 1,000 glomeruli on each hemisphere of the olfactory bulb. And so each glomeruli receive input from one type of olfactory receptor neurons. So the olfactory receptor neurons that are of the same type converge. Somehow they find their way to go and converge onto a single glomerulus. And so these are distributed, scattered in different zones. Now there is 1,000 olfactory receptor gene, and we have almost the same number of glomeruli, but there's the right hemisphere and the left hemisphere. So, so there's a lot of glomeruli, but let's say if we simplify, excuse me, and say that we have glomeruli the same number as the alphabet, like 23 or 26, and if, for example, orange activate A, B, D, F. Banana activate C, M, N, O glomeruli. So which means that each odor can activate different combination of glomeruli. And so this is how, so each combination represent a specific odor. So that's why you can create, I mean, billions of combination with different letter from 26 letter. And so this is how the animal so perceive odor by representing each odor with a map of activated glomeruli. So when so look at the glomerulus, the olfactory bulb as a TV, and where there is a picture. So the pattern of activation of the glomeruli, each one represent like certain odor, a specific odor. Now, if you increase the concentration of odor, you also activate more neighboring glomeruli. So you activate, you become a little bit non-specific. So the way to demonstrate that olfactory receptor neurons of the same type converge onto a single glomerulus, so we use dye labeling, and so it's a certain gene, and you can see that the axons, so they go through tract, and then they converge onto a single glomerulus on the right and the left hemisphere of the olfactory bulb. So even if you destroy the nasal epithelium by a virus or damage or something, the olfactory receptor neurons, they have to regenerate, and at the same time their axon has to travel back to the same glomerulus. So if they go to a different glomerulus, then you perceive the wrong odor. You'll erroneously perceive a certain odor because of the damage, and so that cell has to go back and make synapse onto the glomerulus. So like the taste bud, so similar structures, the nasal epithelium. So first, it's important to recognize that you need some kind of mucus, a thin layer of liquid. So the odor has to diffuse through that liquid or dissolve in it, and then bind to a receptor on the cilia of olfactory receptor neurons. So if your nasal epithelium is dry, you will not be able to perceive odor. Similar thing for taste. So your tongue should always be a little bit moist to have a good functional taste. So this is created by a gland. So glands secrete some mucus. And so we have different type of cells. So first, the main one are the olfactory receptor neurons. So these are the excitable cells. They once odorant bind to them, so it stimulates the neurons. They generate electrical activity, and activity travel through the axon all the way to the olfactory bulb. Now there are other supportive cells. They could also have microvilli, and there are cells that surround the axons to like insulate them, and shielding cell. So if you look through an electron microscope, you see that a lot of cilia on top of the nasal epithelium. And so what's the advantage of having all these cilia? So it's like hair-like structure that have receptor in them. You're actually increasing the surface area. So the more you increase the surface area, you will be able to more capture the signal from small concentration of an odor. So this is very important. So increase the surface area will lead to more responsiveness. Now, a cross section of the cilia, you can see that it has a lot of mitochondria. So I don't know if you remember from other courses that mitochondria, this means that a cell has an increased metabolic activity. So the mitochondria are like the breathing lung of a cell. So the more, so it's like, they have create ATP, and if there's a lot of activity, usually you have more mitochondria. So this usually occur in axon terminal, but it also occur here at the level of microvilli, and you can see that they have also cilia. So it increases the surface area, and so there is a lot of metabolic and electrical activity at this location. Now, the structure of the olfactory bulb. So this is in a mouse, but you're going to see the similarity between the mouse and human. So from the outside to the inside, so you have different layers. So you can recognize them usually by just looking at the slice. By trans-illumination, you'll see different type of layer. So you have the olfactory nerve layer. So this is the layer where you have axon incoming and making synapse inside the glomeruli. So the glomeruli, single for glomerulus, is it's like a module for processing olfactory information. It's like microcircuit in a computer. So you have different of them. And so each one is one processor. So they are spherical in shape, about 100, 150 micron in diameter. And they contain like 10,000 neurons each. So they are one unit. So the first stage of processing olfactory information is at the level of glomeruli. Then you have the epixiform, plexiform layer. So this usually contains mostly dendrite of cell, but it contains some tufted cell. We're going to show this in details. And then you have the mitral cell layer. So the mitral cell layer is the layer that contain the output neuron of the olfactory bulb. So you get all kinds of processing, computer processing. And so this mitral cell, which is much lower in number, they will communicate the other information to the piriform cortex, to other region of the brain. So this is a cross section, and you can see here at higher magnification, this is a different layer. In human, you can see it's very similar, so except it's like a slender nerve. So if you do transverse section, you see the olfactory nerve layer, the glomeruli, the epixiform layer here, and then the mitral cell layer. So in the epixiform layer, you see there are tufted cell. Here you have mitral cell. And then below the mitral cell layer, so there is granule cell. And so the mitral cells, they send their axon via the nerve tract. So it's called the LOT, or lateral olfactory tract. And so this is that connect to other region of the brain. Now, if you look through a Paxinos and Watson atlas of a mouse brain, so you can see the olfactory bulb, so at different level. So they, so when you cut different section, so before we know the functions, they have been labeled like anterior, posterior, lateral, medial. So there's all this kind of labeling of the structure. So again, I mean, here's like more detailed anatomy. So you can, but the idea is to show you, I mean, sometimes the same nucleus have different division, the dorsal, lateral, medial, ventral-posterior. And see here, this is a LOT, or lateral olfactory tract. So here we are talking about the accessory olfactory nucleus. So, so that's to show you how it looked like and how later it connects to the piriform cortex and other nuclei. Now, at the cellular level, now I'm going to show you this more in detail. So if you don't have time today, the other course I'll give more details about how the neurons interact with each other. So the main things to understand here is that there are two types of neurotransmitter, GABA and glutamate. GABA is inhibitory, glutamate is excitatory. And so the neuron, some are excitatory, some are inhibitory. And there is some kind of dendrodendritic interaction. So unlike in other regions of the brain, where usually you have the axon that makes synapse onto dendrite, here you have the dendrite interacting with other dendrites. So the dendrite can act like axon because they receive input, and at the same time, they release neurotransmitter, and so act on other dendrite. So there is this dendrodendritic interaction usually between the tufted cell and the periglomerular cell. So you can see there is dendrodendritic interaction. So one neuron release glutamate, the other neuron release GABA. So this reciprocal interaction. So you have uh, uh, glutamate released from the mitral cell apical dendrite, and you have GABA released from the red cell, the red dendrite, and acting onto the mitral cell. So this is like feedback inhibition. So if there is too much excitation, you need to push on the brake, and so the GABA is released when the interneurons are excited too much, and they will inhibit the excitatory neurons. So they will prevent them from being excited too much. And of course, you have the first input is from the olfactory axons. So this is usually glutamatergic. So it excites both the mitral cell and the juxtaglomerular cell, which release GABA. Now, this activation looks complicated because it occurs in different sequence. So sometimes you have, first you have the excitation, and after the excitation, you get inhibition. So you like stop the excitation. So that you'll see the neuron will burst. So this is show you how we investigate the electrophysiology of this neuron. So we use an electrophysiology setup. And so we dissect the olfactory bulb. So these are horizontal slice from the olfactory bulb. And you can see that, so we put the brain slice into a small chamber, we call the patch clamp chamber, and we submerge it with fluid, and it's circulating. So this is oxygenated cerebrospinal fluid. So we make it in the lab, so it's artificial ACSF. And then, so we can record single neurons with different electrodes. So sometimes I have different micromanipulators, so I can record different neurons simultaneously. We label the neuron with a dye, like lucifer yellow or biocytin. And then we fix the slice with formaldehyde. And then we take image of the neurons. So we can do immunocytochemistry and take image. So we can group the cell into different categories according to their morphology. So you can see there is some type of cells. So here we are looking at cells that inside that are affiliated with one glomerulus. Now we call this juxtaglomerular cell because all the cells are around the circle. They are not inside. So the cells surround the sphere, and they send dendrite inside the glomerulus. So that's why they are surrounding. They are juxtaglomerular. And so the first type is external tufted cell. You can see it extend dendrite throughout the glomerulus. And this type of cell is excitatory, so it releases glutamine. Now the other type of cell is much smaller, and most of the time you don't see the axon. So can you imagine a neuron without an axon? Why? Because the dendrite, actually, they serve both as dendrite and axon. So they receive excitatory input, and then they release GABA onto other cells. Now there is another type of cell we identified. It's called short axon cell. But if you look at this cell, actually it has a very long axon. And so why it was called short axon cell? This is like the first people who discovered it. They didn't have like good labeling tools. So here in our technique, we inject the dye into a single cell, and we see how it diffuses throughout the axon dendrite. So we can follow on the axons for long distance. I mean, when I say long distance, I mean like a couple of millimeters. So this means that the process of these neurons is not restricted to one single glomerulus. It interconnects several glomeruli. So this is like it serves to either increase the excitation of neighboring glomeruli or inhibit surrounding glomeruli. So it can have dual function. So the first person who draw a schematic of how the neuron interact in the olfactory bulb is Raymond Cajal, and he got a Nobel Prize. Now, at that time, there was no electrophysiology, but he was able to guess, for example, how the nerve impulses, so it start from the axons, go and activate the tufted dendrite of tufted cell and mitral cell. And so the impulses propagate from the dendrite to the soma, then from the soma to the axon, then they join the axon bundle, the lateral olfactory tract. So this is before we know about electrophysiology. So he was able to make a good guess of how activity in the brain propagate. And so this is just using a microscope. So before that, I mean, remember we thought about the brain as a box that's complicated. We don't know how it works, but using a microscope, will labeling the cells. This is probably called Golgi labeling, where you only label a small percentage of the cells, so you can follow on their dendrite and process. And so you'll be able to trace the connection between neurons. Now, this is my own schematic. This was part of a review chapter in a book. And so it simplifies a little bit the connection in the olfactory bulb. So you can see the olfactory nerve axons. So sometimes you have thousands of them. So I'm only showing few because to simplify the schematic. And so they make connection inside the glomeruli on the dendrite of tufted cell or mitral cell. And then you have the juxtaglomerular cells. So these are the cells that surround the glomerulus. And so they form part of the processor. It's like a unitary module to process olfactory information. So as we go deeper inside the olfactory bulb, you can see that the number of cells decrease, but their size, the soma size, increase. So basically here, it's like these are the output neurons that send axons through the lateral olfactory tract. So they have to make a decision. Is it what kind of smell it is? So there is fewer number of deciders to decide on the quality and the quantity of the odor. So here you see that there is only 25 mitral cells affiliated with each glomerulus, whereas there are 1,000 juxtaglomerular cells. Now this is another schematic, and it shows, so after recording from different types of neurons, how the neuron interact among each other within the single glomerulus. You can see that the external tufted cells generate bursts of spike. And so these bursts of spike are synchronous among neurons. So if one neuron bursts, the other neuron will burst at the same time. So they coincide. So it's like the entire glomerulus show bursting activity. So it occurs simultaneously. So there's a burst and then there is a pause. There's a burst and there's a pause. And this coincides with the sniffing frequency. So the sniffing frequency occurs at something we call the theta rhythm. And so this is like in animals from 3 to 5 times per second. So they sniff. Now there's active sniffing. So if there's an odor, they will start sniffing at higher rate, maybe up to 8 times per second. Now other neurons, so the PG cells, they are periglomerular cells. They are smaller in size and they are inhibitory. They receive input from the external tufted cell. And how do we know they receive input? There is what's called the excitatory postsynaptic potential. So these are the synaptic activity. And so it's excitatory, it's occurring in a burst. And so they are superimposed event that occur in a burst and a pause, burst and a pause. But here, these cells are receiver. So which means they only fire action potential when there is enough synaptic input to trigger an action potential. So when the membrane gets depolarized enough to reach the threshold and generate action potential. Whereas these neurons are pacemakers. So it's like the heart. They generate activity spontaneously, even without any input. So they generate certain rhythm that's similar to the sniffing frequency. Now the mitral cell are the output, and so they basically buffer the input. So input that is bursting here will be perceived here as like a long-lasting depolarization. So you have some kind of oscillation. So depolarization and then hyperpolarization, depolarization and hyperpolarization. Now, to prove that the neurons are synchronous, you have to record them simultaneously at the same time, so with one electrode in each neuron. So this is require a lot of skills, but and the neurons are too small, so it's quite difficult. But we were able to record some, and only few such recording you can prove what's happening. So you can see that here the blue cell and the red cell, they generate bursts of spike that are synchronous. So they occur at the same time. Now here we record from a blue cell, big tufted cell, external tufted cell, and from a periglomerular cell. So you can see that the blue cell generate bursts of spike, whereas the red cell has bursts of synaptic input. So which means that the blue cell is releasing glutamate in a bursting fashion onto periglomerular cell. So the periglomerular cell probably integrate input from several ET cells. So they're connected probably to 5 to 10 ET cells. That's why they have so many number of synaptic input that exceeds the number of a burst of spike. So they receive bursts of input from synchronous external tufted cell. Now this shows you the connection between the olfactory bulb output. So you see the mitral cell and the tufted cell. So they connect via the lateral olfactory tract to different layers in the olfactory cortex. So there is layer 1A, B, 2, and 3, depending on where the soma are located. So these are olfactory piriform cortex neuron, pyramidal cell. And you can see the connection could be either on the dendrite or on the soma. And there are several interneurons, not just in the olfactory bulb, but also in the piriform cortex. So the circuit can be quite complicated. But something to note here is that the olfactory cortex neuron can feed back onto the olfactory bulb. I mean, the bulb connect to the cortex, but the cortex send back information to the olfactory bulb. And this is so via this pyramidal cell, they send excitatory input to the granule cell. The granule cell are inhibitory neuron, which connect to the lateral dendrite of mitral cell. So there is talk between the olfactory bulb, the main olfactory bulb, and the cortex also send back information to the bulb. So they are interconnected. Now, this shows you how anatomically the bulb connect to different structures within the olfactory cortex. So the piriform cortex, so there are different names here. If you, like to learn anatomy, I don't memorize all of this, so I'm not asking you to memorize them, but just to show you how the main olfactory bulb is connected to different structure within the primary olfactory cortex. And then some of them, they feed back onto the olfactory bulb. So there is a connection back. Now, the accessory olfactory bulb, we said it's like on the dorsal part of the main olfactory bulb. So it's important for pheromone recognition. And you can see there are many nuclei that connect to both the main olfactory bulb and to the accessory olfactory bulb. Now, these nuclei, we're going to learn in other lecture. For example, the locus ceruleus, it releases noradrenaline. Dorsal raphe release serotonin. Diagonal band of, I think there is acetylcholine. So there is like some neurotransmitters that are modulatory in nature. So usually glutamate and GABA are the main neurotransmitter in the brain, but you have other modulatory neurotransmitter, norepinephrine, dopamine, acetylcholine, serotonin, so that modulates the activity of the bulb. And you can tell that if you do immunocytochemistry against different neurotransmitter. For example, here, so you have the choline acetyltransferase, dopamine beta hydroxylase, and serotonin. So if you do immunocytochemistry, you can see that there are projections inside the olfactory bulb of axons that contain this neurotransmitter. Now, what's important here is that the pattern of projection differ for each neurotransmitter. So you can see that acetylcholine, for example, innervates the glomeruli and almost the entire bulb. But for example, dopamine doesn't go to the olfactory, to the glomeruli. It remains mostly in the deeper layer, like mitral cell layer and granule cell layer. Serotonin, you can see it labeled very well the glomeruli, but not the external plexiform layer. So which means that each modulatory neurotransmitter has a specific function in the olfactory bulb. So it can either maybe promote the odor stimulation, or it can suppress. So this is now it show you what are the other nuclei. So after processing the odor information in the olfactory cortex, so the animal need to make a decision. So that's why eventually you have information that's being transmitted to the entire brain, and so to take an action. So this can go to the hippocampus. The hippocampus is a center for memory, so we can remember an odor, so that we can recall later that, oh, this is a nice odor. We can associate it with something good. So there is also a connection with the nuclei that are part of the autonomic nervous system. So you have sympathetic nervous system, which means that you activate high alert. So it's like you increase the heart rate, blood pressure, or you can activate the parasympathetic system. The parasympathetic system is antagonistic to the sympathetic system, so it decelerates the heart, it decreases blood pressure. So sometimes if you smell certain type of tea, you decrease heart rate, blood pressure. So that's why some type of odor are prescribed to relax, so, or to better meditate. So maybe odor of a rose can relax. So the olfactory signal, so it elicits motor activity. If there's an odor that we recognize that is nice, so the glands are excited, so we salivate. So sometimes you go to a restaurant, you smell the food, you start salivating immediately. So it's like your digestive system is trying to get you ready to receive the food. So the enzymes are being prepared, and so so you do this in anticipation of the food. Now, if the food looks bad, you have trash, you start to smell, and it's really bad. So you have nausea, it's like illicit vomiting. It shows on your face that it's unpleasant odor. So it's all of, because the olfactory bulb connect with several structures into the brainstem, where you have salivatory nuclei, you have also nuclei that are involved in gut motility. So it helps in the peristalsis of the GI tract. So the human olfactory system, I said, is much simpler, very small compared to animal. And so we can recognize both pheromone and odor. And sometimes, even if we close the nares and eat food, so you can smell the odor of the food, because the air. It goes through the nasopharyngeal cavity, and so odor can be recognized even if the nerves are closed. Now, there is gender difference in terms of odor sensitivity. Usually women are more sensitive to odor, and especially if they are pregnant. There were some studies that initially they found that the menstrual cycle can be synchronized among female living in the same dormitory. But later, I mean, they found out that this is not really true. The study were not well done. So these are not have been confirmed. So there are some people that have congenital anosmia. This means they cannot smell. Some people normally they cannot smell the natural gas. See, the natural gas you always have to. It's odorless. You have to put some kind of smelly odor, so that when people smell it, so they will know that it's toxic. They have to leave the area. There are some odor, for example, like carbon monoxide. You cannot smell this. So this is if you have like in the chimney some wood that's not burning well, so it will release carbon monoxide, so it can suffocate, kill people around, and they will not able to smell it. So that's why you need the smoke, the carbon monoxide detector to warn you whether there is such a gas. So women can smell better than men. So this shows you the anatomy, the cribriform plate. You see the pore on the base of the skull. So this is where the axon from the olfactory receptor neuron, the nasal epithelium, they go inside the brain. So here we're looking from the bottom towards the base of the skull. And so the pore, you can see them. So this is a possible entry of pathogen, including the virus, but of course they have to go through the cytoskeleton of the olfactory nerve axon. This is more anatomy, show you how the olfactory bulb look like at the base of the skull. So basically, when you look at the base of the brain, you can recognize a little bit the olfactory bulb as a small gland, but then you'll see the nerve. This is the olfactory tract, and so you see how it connects so near the optic nerve and the basal bone. So these are the region of the brain that where the fiber of the olfactory tract terminate. So this includes parts of the amygdala, piriform cortex, and the anterior olfactory nucleus. So the projection of the primary olfactory cortex. So you can see this is the olfactory bulb here, and so it connects to different brain region from one nucleus to another. So it's the hippocampus to memorize the odor, to the orbital cortex, and this is just another way to show the olfactory bulb, how they look like. It's not schematic. So it shows a axon of the mitral cell. So these are the mitral cell. So going through the olfactory tract all the way to the piriform cortex, and so other processing nuclei. Now, there's also the nerve can also bifurcate and go to the other side of the brain. It's called the via anterior commissure. And there are other structures that are important in the perception of odorant. So why we call the piriform cortex? It has a shape of pear, and so this is where you have additional processing of olfactory information. So we talk about different brain regions. So like the amygdala is involved in the emotions. So depending on the perception of whether an odor is good or bad. So there are other regions where you can store information about memory information about the odor. Hypothalamus is also involved in emotion and motivation. So what kind of problem or deficit that occur in olfaction? So as I said, it's not essential to survival. So if we have dysfunction in the sense of smell, so we don't smell anything at all, so we call this anosmic. Now, problem could occur either at two levels, so conductive, when the odorant could not reach the olfactory epithelium. So if you have inflammation in the nose or polyps, so the odor cannot access the olfactory epithelium. Now, sensory neural means you have now a damage to the olfactory receptor neurons themselves. So this could be because of a viral infection, or it can be because there is some problem within the olfactory valve. So it's like injury, traumatic injury, neurodegenerative disease like Parkinson and Alzheimer. So as I mentioned, so olfaction is a good diagnostic tool for several type of brain disease, neurodegenerative disease, and also seizure activity. And this is a clinical case where a woman was smelling odors that do not exist, so some kind of burning odor. So which means that there is something in her brain telling her that there is some kind of odor. So some kind of activity that generated spontaneously within her olfactory cortex. So, and when this woman died, they found that there was a tumor at the base of her brain. So it was like this tumor was growing and in part was affecting the olfactory cortex. So it was affecting her perception of odor. And so it's a big tumor. So this is, as I said, if there is a dysfunction in the sense of smell, it can be sign that there is something wrong in the brain. So it's a good diagnostic tool. So that's why I make sure that you always smell things, your smell is functioning normally. If not, then ask yourself a question, is it a viral infection or do you have something else? You have to do a scan of your brain and see whether there's any neurodegenerative disease. So any question you have? Is everybody here? Say hi.
Hi.
Hello.
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 So I hope I didn't give you a lot of boring information, but it's a very important topic with regard to COVID-19. So I said, so the conclusion is if you've been infected and you lose a sense of smell or taste, I read the recent literature, it's only transient. And so these two sense can regenerate. So you have epithelial cells that can regenerate, and some neuron can still divide and bring back the taste to the normal control condition. And so if you have any other question, just let me know, and or you can contact me by email. Okay?
Okay.
So I'll see you next time.
Thank you.
Okay.
Thank you.
Dr. Syed, you can stop the recording. Are you still there?